One of the great things about being a clinician scientist is that you can treat patients, but also come back to the lab to try to discover more detail about the underlying disease process and also develop new and innovative therapies for patients with intractable disease. I'm a glaucoma specialist, which affects the nerves at the back of the eye that enable us to see. And while there is currently a lot that we can do for patients, unfortunately some of them do progress to blindness or present to us too late to cure. The axons of nerve cells function like a railway system, where the cargo is essential components required for the cells to survive and function. In neurodegenerative disease, such as glaucoma, this railway system can get damaged or blocked. Gene therapy is an emerging technology that can be administered directly to cells that are being damaged. Delivering two molecules that work together can be more effective than either in isolation, and can lead to a more sustained therapeutic effect. This approach allowed us to restore the ability to transport a tracer dye through the damaged axons from the eye to the brain, with potential for vision restoration.
Axon transport is a particularly attractive target because during development, when axons are actively growing, there's an efficient supply of the machinery that they need to stimulate growth. After maturity in the adult, these transport mechanisms are down-regulated, so they're a lot slower. So we figure if we can increase the transport of useful protective or regenerative machinery, then we might be able to repair axons after injury or disease.
So glaucoma is the leading cause of irreversible blindness worldwide, and it's responsible for 4 million cases of blindness and more than that in terms of moderate and severe vision impairment. Gene therapies for glaucoma is a very welcome addition to our potential armamentarium to deal with glaucoma.
One unique aspect of the gene therapy used in the current project was the ability to deliver multiple proteins via a single treatment, and this has a potential to address other blinding pathologies such as diabetic retinopathy and age-related macular degeneration.
As a part of the research, we were testing different model of neurodegenerative disease, which was dementia. After we delivered the gene BDNF and TrkB, we see possible promising data in memory enhancement.
So gene therapy has already proved to be effective for some rare conditions that are caused by a single faulty gene. While this is early data, which requires further investigation, we hope that it will prove to be similarly useful for conditions such as glaucoma or dementia, which are caused by multiple faulty genes and are much more common.